Hello, everybody. I'm going to talk today for December 9th and 10th about the Superbugs lab and also the 122, which antibiotic is the best choice lab. And we're going to just go through what I need you to do on your non-in-person day, the day that you're at home, whether that's blue or gold. So let's get right into it. For 122, for that activity, bring up that assignment real quick. 122. Where I wanted you to fill this out. Just a reminder to make sure that you have this turned in by Thursday the 10th and get that submitted to Canvas. Please ask questions when you're in class this week, Wednesday or Thursday for that. That is going to be turned in right here. So then on to the Superbugs lab data. We're also going to be analyzing the antibiotic, our portion of the 122 lab in class on Wednesday or Thursday, depending upon when you're here. We're going to look at our results. That's going to take a good amount of time because you're going to have to make drawings of your results, do some measurements, and write out a data table. That will also be a part of your submission. So you're going to add a picture of your results to your 122 workbook. So I'll talk more about that in class. So that's going to take a lot of time. Additionally, 123, we're going to be working on setting up some plates in class where you're going to be using bacteria cultures to make confirmation plates. So we have two different bacteria, and I'll talk about that in a minute. And we're going to confirm that they're resistant to what it says they're resistant to. So that brings me to where we're at here in the Attack of the Superbugs. I went through the notes on the last week, or on the last lesson, where I talked about Streptococcus and how it's become resistant to antibiotics over time. You see other types of infections that we talked about, ear infections, pneumonia, skin infections, and so on, that are becoming resistant to antibiotics. So in this lab, our goal is to look at how DNA can be transferred from one bacterial cell to another. Bacteria reproduce asexually, so really there should be little transfer and change from one bacteria to another, or trading of genetic information. It doesn't happen very, very easily in organisms that reproduce asexually. So, but if one bacterial cell is resistant to an antibiotic, it has the ability to transfer that to another cell, and that's how it can spread relatively easy, right? And so then you get this whole army of superbugs that are going to be more difficult to kill. All right, so when we go, we're going to scroll down here to under step 4. All right, we talked about bacterial cells in 121, okay? And just to kind of review that, 121, remember this, the bacterial cell structure, and you have a plasmid and you have DNA. Plasmids are DNA, and the nucleoid is DNA. This is the nucleoid, remember, is the main region of genetic information for bacteria. Plasmid is kind of extra information that can be used to help bacteria survive, or just randomly happens to be there. So antibiotic resistance can be on the nucleoid, and it can be on the plasmid. Okay, so that's important to know for what we're doing for 123. So if you look through here, this paragraph's really important, and I'll bring up the 123 document also. Remember that 123 document? I passed it out to the gold people on Tuesday. Blue is getting it on Wednesday. This Attack the Superbugs lab data, and essentially we're done with this first part, right? So now we're looking at bacterial strains and confirmation plates and predictions. So here you're going to describe each bacterial strain you'll be using during the lab. Now, strain basically means version. It's not like when you pull a muscle. It's not like when you put spaghetti in a colander where you're straining it. No, the bacterial strain is a version of the bacteria. So we're using 2 different versions of E. coli bacteria for this lab. And for each one, you need to describe whether it's resistant to ampicillin, which is one antibiotic, or streptomycin, which is another antibiotic, and where the resistance is found for each. And really the 2 options are the nucleoid, which is where the main DNA is, or the plasmid. And if you read this sentence, you should be able to get that, right? So You can have DNA in the chromosomes or the nucleoid. You can also have DNA in outside the nucleoid in the plasmids. So it talks right here in this sentence. So take that information and describe that in this sentence right here. Basically use that to describe E. coli strain 1 and E. coli strain 2. All right, then you're going to make some predictions, and I do want you to, it says date your prediction. That doesn't mean you ask it out. But date your prediction means I want you to write your date that you predicted these things on here. I want to make sure that you did that before you actually, once you have this done, you know, we may predict this before, or we'll do it after we do the lab. Basically, before Friday, you need to have predicted this because you'll see the results Friday. All right, and so what we're going to be doing is you're going to have strain 1, you're going to have strain 2, okay? And so you're going to take strain 1 for each of these plates, and you're going to streak it, okay? Swap it back and forth. Strain 1 on all 4 of these plates. Strain 2 on the other half of each of these plates, okay? And one plate is just LB agar. LB stands for Luria broth. It's just essentially, it's like hay for horses, right? It's the stuff that they eat. It's what they love. It's what bacteria want to grow on. And so you have, in this case, the agar is just plain agar, right? There's no antibiotic in it. This one, there's streptomycin antibiotic in it. This one, there's ampicillin antibiotic in it. And this one has both strep and ampicillin in it, okay? So, based on the information that you're reading 1, 2, 3, make a prediction as to whether strain 1, is strain 1 going to grow? Which plates will it grow on? And shade it in. If it's going to grow here, you would shade in this half. Here, you're going to shade in this half. If you think both would grow on this plate, you would shade in both halves. If you think neither would grow on this plate, you would shade in none of them, okay? So that's a prediction that you need to make. And I'll be checking. I do want to see those predictions on Friday before you do the lab. This we're going to skip for now because these are the results that we're going to look at on Friday, okay? We're going to just basically state whether they grew or not. So before we get to that, though, I'm going to have you go ahead and do one more thing for research, which is you're going to look at different methods of antibiotic resistance. And so for that, you scroll down. This is all the procedure that we're going to follow in class. I would like you to kind of look and see and read through this procedure as well. So that's the other part of this, is to read through all this procedure. Okay, it shows you how to streak the plate. And I'll demonstrate this, but it'd be very helpful if you did that in all the classes. So you're going to basically follow E. coli 1. Half the plate will have E. coli 1. Half the plate will have E. coli strain 2. And then we're incubate them for 24, in this case 48 hours. I will take them out after 24 hours, but and then you're going to put those observations in. And this is where I want you then to look for the different methods of antibiotic transfer, which I believe is up here. Yeah, so then we're going to scroll down to basically step 69. All right, and so it's a little bit farther down. There's a video here from the Food and Drug Administration over antimicrobial resistance. And we're going to look at how, so these things become resistant, how they become resistant. So I click on this. It talks about there's an animation of antimicrobial resistance. Right? So if you click on this, there's different versions of it. And watch this video. It's about 9-minute video, and it basically goes through each of these and kind of defines each of these. So you'll want to just look for the first. I'm going to show you where it starts talking about some of these mechanisms of antimicrobial resistance. Definitely want to watch the first part, but then you get into the different mechanisms, and it talks about the 3 types: mutation, destruction or inactivation, and efflux. So those are the things. So this is where you would see that in the video. And then you would watch that, and then later it talks about genetic transfer. So from about, basically from the beginning of the video through about 6:18 is where you'd want to watch. We're going to cover these other methods on later in the week, either Friday or maybe our last day next week. All right, so then use the information that follows in this video. This is what I want you to be doing either Wednesday or Thursday when you're at home. is describe and maybe add a draw, and also add like a drawing. So you need to write a description and a drawing for each of these, and add that to the document. So just a reminder, on the day that you're not here, you need to be describing the difference between the strains, make a prediction, and look at the difference between mutation, inactivation, destruction, and efflux. And these are, once again, are how does that initial antibiotic resistance happen? All right. So how does that happen in the first place? Like, what are the different ways that bacteria can come up with, or there are different sort of counterattacks that they have to prevent the antibiotic from working. So have that filled out on the 1, 2, 3 paper when you come to class Friday, all right, so that we'll look at the confirmation plates. We'll have basically make like a mixed plate, and then the following week, that'll be Tuesday, Wednesday, we're going to finish this lab out. There will also be a data, there will be a summary that I'll be assigning that will be due at the end of the week. All right. Additionally, we may work on this one. There's a 1, 2, 4 quick lab that we'll be working on in class as well. So that's pretty much it. And make sure that you turn in the 1, 2, 2 antibiotic lab once you've gotten the results from class. Make sure you turn that in the Superbugs lab, and then you'll be good to go. So let me know if you have any questions via email, and I will make sure to address this as best I can in class. Thanks.